It's been a very long day. Yes, probably many of you are already tired, okay? But anyway, it's the first day, you're still fresh. It will be harder probably on Thursday. Anyway, so the question is like, today you have been introduced to probabilistic AI. Celia this morning gave you like the basics idea. Andrew were like a bit farther with the Bayesian workflow, but so far you have been exposed like mainly like toy examples, okay? Because of pedagogical purpose, like simple models and so on. And then today we wanted to give you a hint about like the potential application of all these techniques in very relevant field. And this is because we invited Wouter, okay? He's a professor at the computer science department at Copenhagen University. And he's been working a lot on applying probabilistic machine learning, probabilistic AI in bioinformatics. So I think you will enjoy like the potential of all these techniques in a very relevant application field. Okay? Yeah? Thank you.
Thank you for the invitation. So I was invited to give a talk about probabilistic machine learning in bioinformatics, as the title here says. And so there are two problems with this. One is that bioinformatics is quite a big field. This is, if you look up on Wikipedia, what bioinformatics is, it's at least all of this and new technique, new sort of methods coming out. Every time a new experimental technique is, someone comes up with a new experimental technique, this list will become longer because it's a lot about processing biological data. So that's. And if you then look at the topics in probabilistic machine learning, here I just took the topics from this week's summer school. This means that there are quite a lot of sort of points to go through in an hour-long talk. So in addition, there's the other problem, and that is that this is the first day of the summer school, and a lot of these topics you haven't yet been introduced to. So talking about them in great depth is maybe not the right thing to do at this stage. So what I've decided to do is to take one topic from bioinformatics, this particular one, which is about amino acid sequence modeling. And then I'm going to try and sort of discuss this topic from various methodology angles. So I think if I get around to it, I'm going to talk about variational inference, a little bit deep generative models, a little bit about diffusion models. I might mention a little bit about Monte Carlo. I'm going to talk a little bit about Gaussian processes, all sort of centered around this one problem, which is amino acid sequence modeling. And this has, and I'm going to try and keep it, it's been a long day, I'm going to try and keep it fairly light so that we don't fall asleep. So the advantage of this particular focus is that I only have to introduce one biological topic to you. I will need you to bear with me for 3 slides. I'm going to talk about 3 aspects of amino acid sequence modeling. So the first thing that you will need to know about proteins, and you might remember this from high school, is that proteins are chains of amino acids. There are 20 naturally occurring amino acids. And because there are 20 of them, they're conveniently represented as letters from our alphabet. So we can really represent a protein simply as a sequence of letters. And this means that basically when I talk about modeling protein sequences, it's a sequence modeling task. So that's the first item of knowledge you need to have about biology. If there are any questions, please ask them as I go through these 3 pieces of information. You will definitely need to know all 3 in order to understand what's going on. So that's the first thing. The second thing is that although proteins are sequences, we should have in the back of our minds that these chains of amino acids, they fold up, many of them, most of them, fold up into these compact 3D structures. And this means that although they are sequential in nature, they also have sort of non-local interactions that come through the way they're structured. So they fold up onto each other. And we typically represent them in this cartoonish fashion, but this is just the chain that I showed on the previous slide, and then we have these cartoonish ways to represent particular local structural motifs here. This is a helix, for instance, and this is a strand. Okay, so second piece of information, sequences encode structure. There is proteins fold to 3D structures. And the third essential piece of knowledge we need for this talk is that proteins are related through evolution. So if I look at a particular protein, a human protein, I can find the equivalent protein in a mouse, for instance, and that protein will have the same shape and it will largely have the same function, but there will be minor differences in the amino acid composition. So they will typically still fold to the same structure, maybe with small variations. So here we see, for instance, the same protein in a slightly different structural representation. They look they have this similar overall fold, but if we look at sort of charge distributions, they might be slightly different. And depending on how far we go away in terms of species, these similarities will be smaller or greater. Okay, so those were the three things we need to know about proteins and amino acid sequences. Okay, so let's dive in. So there will be two parts to this talk. The first is very sort of basic modeling of these sequences of amino acids. So I'm going to talk you through increasingly complex ways that we can model these sequences of amino acids. And the goal here is to illustrate that even though the problem is fairly simple, and we can, I hope, at least at the abstraction level that I've explained it to you now, we sort of understand what proteins are as simply sequences of 20 different amino acids, that the modeling choices are quite critical to the performance and the sort of the downstream applications that we can obtain with these models. Okay. So the simplest thing we can ask ourselves the question: if we wanted to model the probability distribution of all the protein sequences that occur in nature, how would we go about doing that? So we imagine we have sequences of this type here, and we want to model the distribution over these vectors of amino acids, where the protein length here is L, so we have a number of amino acids. We want to model this joint distribution. So the simplest, or the first question we have to ask ourselves is, how do we even do that? As I just said before, if we have proteins from two different species, they're similar, but for instance, in mouse, maybe there are a few additional amino acids at the beginning of the sequence. Sometimes there might be an extra loop somewhere. So it's very difficult to talk about this A1, for instance, the first amino acid in our sequence, as having sort of an absolute meaning, because I can add a few extra amino acids to the beginning of the chain, and then A1 will mean something else. So it's very difficult to talk about absolute positions in a protein sequence. But one trick that bioinformaticians sort of early on figured out was to use alignment to get rid of this problem. So the idea is that you align sequences in such a way so that equivalent positions in the different sequences, so these are different sort of, imagine proteins from different species here, the rows, and we place them such that equivalent positions are in the same column, such that if we imagine this 3D structure, and imagine a particular spot in this 3D structure, that will be represented, all those equivalent amino acids will be located beneath each other in this column, such that we can see that if a particular amino acid is the same in all these different species, if it's evolutionarily conserved, that we can easily see this in this alignment. So that's a trick we need to do. And in order to obtain this alignment, we will need to introduce gaps, because as I said, some proteins might have a loop, an extra loop. In this case, this protein here in the top row has an extra loop here, and in order to then make the alignment work, we need to introduce gaps in all the others to make room for this loop that this particular protein has. Okay, so that's a trick to pre-process the data such that we can model it more easily. Once we've done that, we can now phrase or write up our first model, and the simplest model we can come up with is to assume that all the positions in our alignment are independent. So we're going to factorize this joint distribution by the individual positions. So we're going to say the joint distribution over the amino acid sequence is going to be the product of the distributions at each individual position. Okay, so we'll get back to whether that's a good assumption or not. But the idea is that, or the motivation is that this model we can easily fit, right? Because we can just look at our alignment here. We have all this sequence data, and you should imagine we have hundreds, if not thousands, of these sequences. And we're just going to process each column independently, and we can do this just by counting, right? We can just count how often does alanine occur, how often does lysine occur, and so forth, and then normalize, and then we get a discrete or categorical distribution over the 20 different amino acids at that position, and then we do the same for the next position, and so forth. And then we have a model of the protein sequences in such an alignment. All right. So is that a good assumption to make, that they're independent? We could also ask in a different way, do we see correlations in proteins between sites? And unfortunately for our model assumptions, the answer is yes, we do actually see quite a few correlations. We can actually already see it here in these local structural arrangements, this helix, for instance. There will be specific requirements for the type of amino acids that appear in such a helix. So in order to form a helix, you'll have particular constraints of which amino acids you can have next to each other. So there will be correlations between neighboring amino acids. And then you can ask, okay, is that the only thing? Because maybe we can deal with that. Are there any global dependencies as well? And the answer is yes. Actually, we also have global dependencies, because if you look at the structure like this, there will be places that are far apart in the sequence but are close to each other in 3D structure, and where you can imagine that if you make a mutation here, if you change the amino acid here, that it will affect the amino acid on the other side. So we don't only have local correlations, but we also have, in terms of the sequence, long-range dependencies along the sequence. So our independence assumption is not very good. So the question is, can we do better than that? And the answer is yes. So one thing we can do is that we can say, okay, we know that in our initial model, the model we just wrote up here, after training this model, we know that after we train the model and look at the statistics for each column, we know that they will be correct. So we know that our model will fulfill have the same distribution at each position as the training data, because it's estimated to have that property. So what we can do now is that we can write up a model where we say, not only do we insist that the frequencies at each column are exactly as our training data, but we also want all pairwise frequencies to be the same as the training data. And there are actually many models that will fulfill that constraint, but the maximum entropy model, so the model that makes fewest assumption, is this model, which is called the POTS model, where you end up having contributions for each of the positions as we had before. So this is the same model as before, basically, but now we add this additional, so we have contributions for each pair. And we sum over all pairs here, we sum over the length here. And then in order for this to be a properly normalized probability distribution, we will then have to normalize by this 1/z normalization constant. This is called an energy-based model, because these components here are really energies. They're not normalized probabilities. So these are energy contributions that we sum up to get a total energy, and we then need to normalize that to get a probability. This normalization constant will in general be very expensive to calculate, because in order to normalize this, we would need to sum over all possible amino acid sequences that we can imagine ever observing. And since we have 20 possible amino acids at one position, and we have a length of L, that would be 20 to the power of L possible sequences. So this is an extremely large sum to have to compute. So this makes this direct sort of modeling, this direct calculation of the likelihood intractable. But for this particular case, it actually turns out that there's a shortcut. You can actually get a very good approximation of this model. You can estimate it reliably by using something called a pseudo likelihood. I won't go into the details here, but just to say that you can actually estimate this particular model. And the advantage of doing this is that we now have a model where we both have this frequencies for each position and all the pairwise frequencies correct. Is that not only does this give us a better model in terms of likelihood, presumably, but we can also actually interpret this model. We can analyze which positions in our chains, with this long chain of amino acids, which positions actually demonstrate correlations between them afterwards by inspecting sort of which have non-zero or non-trivial sort of energy components between I and J. So we can look at this. And this is something that people had been looking at for years prior to this to find a good way to do this, because for a long time there had been this idea that if you just had enough sequences, like thousands of sequences, and you repeatedly saw that particular sites had correlated mutations, so that at site 10 and at site 37, at position 10 and 37, you would see a correlation appearing again and again at the same time, that would give us some information that these two positions would be close to each other in space. So in the 3D structure, these would be close to each other. And one of the big sort of challenges in bioinformatics for the last 50 years or longer has been to try and predict 3D structure from the sequence. And this has been, we've been working on that for years and years. And so the idea that you could extract some information about the 3D structure from looking just at a whole bunch of sequences, that was really something people had been trying at for a long time, but hadn't sort of figured out the right way of modeling it. Because if you just look at correlations between all pairs, you will, if in situations like this, you will find that A and B here, for instance, correlate, and that A and D correlate, and that D and C correlate. But you would also find a correlation between C and B here. And it was impossible until this point to distinguish between sort of direct correlations and indirect correlations. And this means that it was a very sort of noisy analysis, and you didn't get very good contact predictions. But with this model, because we're optimizing it in sort of one sort of consistent model where we have where we're normalizing everything to give to get a sort of a joint distribution, we actually separate the direct interactions from the indirect interactions, and so we were able to calculate these contacts. I will get back to later in the talk why that matters, but for now let's just remember that we can now do this. Okay, so the next question is, can we do even better than this? So we had one simple model here, which focused on the individual statistics here at each site independently. Now we just wrote up a model where we looked at the pairwise factors, so that the pairwise frequencies we observed in the data set were preserved in our model. Can we do better? So one obvious choice there is to consider whether we could, for instance, go to instead of pairwise, whether we can look at 3 positions at once, or 4 or higher orders. And that turns out to be extremely expensive. We don't have enough data typically to actually train these models. But one thing we could do is to introduce a latent variable, and then say that we want this latent variable to model sort of richer covariances among sites, which can be pairwise or higher order covariances. And that we will then assume independence conditionally on set. So we'll move all the difficult sort of correlations between sites, we'll move that to the set latent variable here. So this I wasn't here this morning, so I don't know if you already talked about mixture models, but if this had been a discrete, if Z was discrete, this would have been a standard mixture model. But right around this time, what was really popular around this time was the variational autoencoder, where you can actually, which showed that you can estimate continuous latent variable models efficiently. And I know that this is on the agenda for variational inference, I think, tomorrow, and latent variable models or deep generative modeling on Wednesday. So you'll hear much more about this, but the basic idea is that we now have this latent variable model, which we have not observed, and that we, through a number of layers, that's where the deep generative modeling comes from, we then ultimately predict this distribution over the 20 amino acids for each position. And as you'll hear on Wednesday, in order to train this, you then need to estimate an encoder also as a variational approximation, because you cannot. So for a discrete mixture model, we could have used EM easily to estimate this, but for the continuous, the VAE, you end up optimizing this elbow, where you simultaneously optimize an encoder and a decoder. Much more on that Wednesday. All right, so we've now seen this progression from sidewise to pairwise to a latent variable model that in principle can capture higher order correlations as well. And so why did we even want to do this? So I started off by saying, let's model this distribution over amino acid sequences, and you all trusted me that eventually I would probably give you some point of the whole, this whole exercise. So there are two things we can potentially do once we have such a model. One thing is that we can generate new sequences. We can generate random protein sequences if that's what we want. That's actually interesting as well, but I won't talk about that right now. But the other thing is that we can assess how likely a given sequence is. And the question is that useful? Is that a useful thing to do if someone gives me a protein sequence that I can say, yeah, that's a pretty good sequence, and if I get someone from somewhere else, then that's maybe a pretty bad sequence. The ability to distinguish between good and bad protein sequences, is that a good thing to be able to do? So what does this probability really mean? So we know that over millions of years, evolution has selected these protein sequences. So there have been, evolution has tried lots of sequences, and many of them didn't work, and then organisms died. So the sequences that are left that we can see now are heavily sort of selected to be good proteins. And this means we expect that, or we know that high probability, according to our model, is correlated with high evolutionary fitness. At least that's what we would expect. And this means that we would expect that we would be able to see a correlation between the likelihood differences if we take, let's say we observe some protein sequence in nature, and then I make a few I mutate the proteins in a few places in the protein. If I then evaluate the likelihood of the original sequence and the new sequence and look at the difference in likelihood, I would expect that to correlate with the difference in fitness of this protein. And it turns out that you can actually do these experiments, and people have done them, DMS or MAVE experiments. And over here, it's difficult for you to read, so I'll just tell you have to trust me. So here there are a whole bunch of experimental data sets. So for each of those, what the experimentalists did was to take a protein, create a whole bunch of mutated versions of that protein, and for each of them measure, for instance, the stability of the protein or some other sort of functional aspect of that protein, how well it functions. For each of those, you could then create a scatter plot where you plot the actual experimental fitness, change in fitness, and then on the other axis we can plot our predicted change of change of likelihood according to our model, and then we can calculate a Spearman correlation coefficient, and then for each of these we can plot these Spearman correlation coefficients and we can see what we get. So we see that in general we do find quite strong correlations, at least for some systems, between the likelihoods that the model assigns and the evolutionary fitness. It differs a little bit from protein to protein, how strong this correlation is. And the other thing we can see here is that we have a yellow dot and a black dot and a gray dot here for each system. And these are exactly the three different types of models that I presented to you before. So the gray dots down here are the site-independent model. The black dots are the ones that included pairwise interactions. And the yellow dots are the latent variable model results. So we see that by doing this, making an increasingly complex model, we actually gained something. We had a better description, our likelihoods are better, but also the correlation to something that's actually sort of a real biological, that gives us real biological insight is higher for these models. Okay, so likelihoods are useful and we can use them to distinguish good from bad proteins to some extent. So the next question you could ask is then, okay, we know that there are quite a few genetic diseases out there. And some of them are quite rare in that we don't have a lot of statistics for them. But we have databases where they've been carefully annotated, at least all the ones that are not so rare. We have quite a bit of data on whether mutations are believed to be pathogenic or benign. So with such a model, you could ask the question, you could imagine that you would expect the model to be able to differentiate between the benign mutations and the pathogenic mutations. And so indeed, you see that if you do this sort of across all these proteins, if you plot the evolutionary index is just this difference in likelihood or the likelihood ratio that I just talked about. And if you then color them whether they're pathogenic or benign, you see that there is indeed sort of a separation between the benign and the pathogenic mutations. And this is really quite interesting because, as I said before, there are a lot of genetic diseases are rare disorders where we don't have enough statistics we don't know all the mutations for a lot of these diseases. But now we have tools so we can use this sort of probabilistic modeling to make statements about, to make predictions about whether we believe a mutation is going to be pathogenic or benign. All right. All right, so I wanted to just give you, so I'm slowly working towards the end of part one. I want to give you two perspectives on this simple modeling task of modeling distributions over sequences. The first perspective is you could ask whether, so by perspective I just mean sort of ongoing work in this or sort of potential impact that this has had. And the first perspective is that you could ask whether, are we done now? Is the VAE sort of the end of the line? Is this the best model we could imagine for this particular purpose? So one potential problem is that if we look at this latent variable model, what we're basically doing is that we still have a similar factorization as the very, very simplest model I presented first, which is we're basically just factorizing so that the distribution of each site is independent. It's just now conditionally independent. It's conditioned on this Z variable. So, but once you've chosen a particular value of Z here, then the distribution of the 20 amino acids here and of the 20 amino acids here will be independent given that choice of Z. And this means that we're asking a lot of this latent variable. It has to model all the complex correlations in the sequence. And the output distributions here, so once we've chosen a Z, then the distribution of the individual amino acids will probably be fairly peaked because as soon as you allow yourself a richer distribution in the output distribution of the 20 amino acids, then you lose the correlation to the other sites. Then you cannot model that. All the correlations have to be in this latent variable set. So one obvious question you can ask is whether you can spread this responsibility of the latent variables, whether you can spread that responsibility over multiple latent variables. So creating sort of hierarchical latent variable models like this. And we and others have looked into this, and especially in the image domain, for many years, variational autoencoders were not very performant because they generated blurry images. And ultimately, that problem was solved by introducing sort of deep layers of sort of these hierarchical structures in VAEs. And so you could ask whether we could do the same for sequences and how deep we can really go. And it turns out that as you go deeper in building these hierarchical models, it becomes difficult to estimate this encoder that you need to get these VAs to fit well. And so there's a limit to how deep you can make them. But you can imagine, so there was an insight that if you let these hierarchies become bigger and bigger, then eventually, and you sort of share parameters, then this model class corresponds to the model class of diffusion models, where you make the additional assumption that your encoder doesn't have any parameters. So where you stop encoding or stop estimating the encoder and make that a deterministic noising process, then you get sort of the equivalent of this hierarchical VAE. And so people are now pursuing this further, and there are some interesting results now where people look at sort of probabilistic models based on diffusion, sort of discrete diffusion models, showing that you can generate even higher quality protein sequences. So in our hands, you cannot, at least what we've seen so far, you don't get the same quality of likelihoods with these models yet. So there's still work in progress, but just to illustrate that still in the year 2023 or 24, people are still working on improving these types of models to become even better. The other perspective I wanted to give was, I promised that when we, in this slide early on, that I will talk a little bit more about the impact of this. So the fact that we could now, with these models, we could separate direct from indirect context, and for the first time estimate which positions in a protein chain were in contact with each other. This started sort of a revolution where people became increasingly successful in predicting aspects of protein structure by processing sort of contact maps through deep neural networks to make them more and more accurate, to give more and more accurate descriptions of which sites in the proteins were in contact with each other. And ultimately this led to, with a lot of engineering and other insights, it led to this AlphaFold procedure, which is built very much around the idea of taking multiple sequence alignments, extracting which things correlate, and then ultimately using that to predict 3D structure. So this simple insight that we had, this modeling choice of modeling frequencies versus independent sites, sort of ended up making it possible to predict 3D structure, which has had a completely sort of fundamental effect on the whole field of structural bioinformatics. For many, many years, structures were extremely rare. We have lots of protein sequences, but in order to get a structure, you would need to spend, I don't know, half a year in a lab to solve it. So the gap between the available sequences and the available structures was just growing and growing. And you really needed a structure to be able to develop drugs and things like that against a protein. So this has had a huge impact, this simple insight, leading to this AlphaFold. All right. Are there any questions to this first part before I continue on the second part?
Yes, maybe it's like you're pointing there, it's high confidence, low confidence.
Yes. All right, so one of the reasons that AlphaFold ended up being not just an academic success, but also a practical success in the sense that biologists started using it on par with experimental structures, was that in addition to predicting the 3D structure, they also trained an additional model to predict how confident they were about this prediction. And this was not sort of the types of model, the types of uncertainties I think you will be talking about in this course are primarily sort of epistemic uncertainties, sort of uncertainties about the model itself that we get from a Bayesian analysis. But this was just a complete separate model that during training estimated how likely are we to get these things right or not. So it was a bit of a mixture of aleatoric and epistemic uncertainties, so reducible and irreducible sources of uncertainty. So it will predict for each position, it will predict the certainty that it's predicting that position with, but certain parts of a structure will be intrinsically disordered in the sense that they don't have any structure. And that will get a low confidence score simply because it will vary a lot in the data set. And other parts might be uncertain because it just doesn't have a lot of data. And these two things are mixed together. But it's still a super useful tool for practitioners to have this available. So that's, yeah.
The alignment
That's a super good question. So the question was whether the alignment problem was solved. So remember at one of the very first slides, I said that we started out by aligning our sequences. So some of these later techniques, such as this diffusion model that I had on the previous slide, one of the latest techniques, they actually don't use alignment anymore. At least for one of their models, they don't use alignment. So they try to estimate based on sort of attention mechanisms rather than doing an alignment. And you can get quite far with that, although it depends on how many sequences you have available, whether you want to learn that from scratch or whether you want to sort of put it in as a pre-processing step, this alignment. Your actual question was whether if you want to do an alignment, whether you should do it probabilistically. I think we've looked into that a little bit. It's in principle, yes, but it's difficult to get very sort of much impact out of that. It seems that optimizing that part is a pretty sort of a good approximation, but I'll be happy to discuss that further afterwards. All right. So I still have a couple of methodological parts to get through. So that's the second part of the talk, is talking about this. If we then, I talked about before that we can take a protein sequence and then we can judge whether it's a good or a bad protein. But, and I also mentioned that we can use these probabilistic models to generate new proteins, and whether that's, so the question is that ever useful for anything? And to answer both those questions, there's a big interest in generating proteins that work differently or better than the ones we find in nature. So the question is, can we use probabilistic machine learning to improve proteins? And I'll just motivate this by talking about this one particular protein, which is called green fluorescent protein, GFP. It looks like this. It was originally extracted from jellyfish, and it has this interesting property that if you shine light on this protein at a bluish wavelength, then it illuminates in a greenish wavelength back to you. So here's a particular, I don't even know what this is, animal, insect, water insect, that has this property, that has sort of this protein expressed. So if you shine light on it, it glows up like this. This has turned out to be super useful in biological experimental setups where you can make this expression of this protein dependent on something you're interested in biologically, and then you can use that to detect whether something interesting is going on in your experiment. And actually, it's so interesting that the Nobel Prize was awarded for this discovery. But it also means that it's very interesting to try and tweak this protein to be better at fluorescing, and to fluoresce at different wavelengths so that you have different colors to your exposure when you're doing these experiments, so you can separate between them. So over the years, there have been multiple sort of variations of these naturally occurring proteins to make them better than the natural occurring ones. But this is just one example. In general, there's just a lot of interest to optimize also enzymes to do different things. For instance, there's a company here in the Copenhagen area working on Novanestis, working on optimizing enzymes, for instance, as detergents in washing powder or for degrading plastics and things like that. You can imagine all kinds of scenarios where you have a wild-type protein that does something that you're interested in, but it's not optimized in biology to do that particular task very well, so you can then try to find better variants. So the typical setup will be that you have some parent, which is just some naturally occurring protein sequence. And then we do mutagenesis. That just means we introduce a bunch of mutations. So we get this pool of sequences where we've made some changes to a few amino acids. Then we have some screen where we can then measure, is this a good protein or not? And then eventually from that screen, we then pick the best ones, and then we mutate them again. So that's sort of the classic setup of how you could gradually optimize towards better functioning proteins. But of course, now in the age of machine learning, we would like to change this setup a little bit. So we'd like to be able to say that from this variant pool that we then experimentally screen, we would like to use this information to learn something about which variants are good and which variants are not so good, such that we, from our machine learning model, could then predict better variants in the next round of experiments. So there's sort of an active learning kind of loop here where we make some proposals for the next round of experiments, and then we get feedback, we update our model, and so forth. So that's the idea here. So the natural sort of methodological component or natural method to use here is called Bayesian optimization. And the idea is that we imagine, so this is, we're basically going to do this. We want to fit sort of some function to our observed experimental data points. So let's say here, the dotted line here is the underlying, the real fitness landscape, which we don't have access to. And then we make experimental observations. So at a few particular points here, and here, for instance, we know what the function value is. And then the rest here is sort of the black line here is then some interpolation between these points. So this is the black line is then our best bet for what the fitness landscape looks like currently. And then we write up another function, which is called the acquisition function. And this acquisition function says that based on our current best bet of what the fitness landscape looks like, we call this a surrogate function, what would be the most beneficial places to explore further? And there are different acquisition functions. But often they are some mixture of taking the function values and the uncertainty into account. And so, for instance, here this particular acquisition function has its maximum here. It says this would be a good place to explore in your next round of experiments, because here you have potentially, given the uncertainty and your current estimate here, this is a likely place that the optimum might be. So you do this over time. You then make the observation. You update your surrogate function. And you gradually sort of navigate this search space to become closer and closer to your optimum. You eventually find your optimum to be up here. Okay, so in order for us to be able to do something like this is a very sort of appealing setup, we would need some distribution over fitness landscape. So we need some distribution over functions here. And this is what I just want to talk about here at the end is a very natural technique for modeling exactly this problem of what we have we need a distribution over functions, our Gaussian processes, which you will hear about on Friday, I think. And the idea is that originally you don't really know much about the functions, so these are just examples of possible functions. But then gradually, as you then have experimental observations, you will sort of narrow down this function, so condition them on that they have to go through a particular point. Whenever you make an observation, you know that the function value at this particular point is this. Here we have some remaining uncertainty here, which is the aleatoric uncertainty, the sort of the irreducible uncertainty, which you can change to make the noise larger or smaller. But this is the sort of the core ingredient that we need in order to model these fitness landscapes and sort of update them in a Bayesian fashion as we acquire more data. So this is a whole topic in itself. You'll be going through it in detail. So I just wanted to make the protein-specific connection here, because a lot of the time, GPs will always be presented most of the time in this 1D scenario. But for proteins, we are most definitely not in 1D. So as I said, a protein is a sequence of amino acids. You can have 20 at each, 20 different values at each position. So you can represent it in a one-hot, or you could choose some embeddings, but typically you will have hundreds or at least or hundreds or thousands of dimensions to represent a protein in as the input for your regression. So this is potentially sort of a big problem for GPs, as you'll hear later on. So the questions I think we have to ask ourselves when we work in this field is, can we actually can we model fitness landscapes in hundreds or thousands of dimensions reliably? Is that something that actually makes sense? And so what is the accuracy we can expect? And we saw that in this Bayesian optimization loop, not only do we need sort of estimates of the value of the fitness landscape, but we also need some notion of the uncertainty that we have. And can we get reasonable uncertainties in high dimensions? And so we have looked into this a little bit, and I want to just tell you about this illustrative case which illustrates why it's sometimes a little bit difficult to work with real data. So we wanted to work on this Bayesian feedback loop and small data regime where we get data in iterations and when we can predict the best candidates for the coming iterations. So we started this collaboration, and they sent us this data set. And I don't know anything about the data set because it's a secret. I just got this plot. But we tried just to take this data set. We didn't really have any metadata for this data set. We just tried to fit it some standard regression method on this data set. And we got a Spearman correlation of 0.94 on this data set. So that was fantastic, problem solved. But the problem is that we knew that these were quite noisy data sets. And so we really didn't expect to get this level of performance. So something fishy was presumably going on. So we then looked a little bit deeper, and we looked for each of the variants that we were making predictions on. How far were they away from the original, the protein we started out with, the one without any mutations? How far were they away from that? And here's a plot where we show the points, the size of the circle is how far we're away from the original. And we see that as we get further and further here along the x-axis, our points become larger and larger. So it seems that the fitness of this protein just keeps on growing the more mutations we make. Now, you might not have a great biological intuition after the 3 slides I gave you about proteins, but that would be an extremely surprising result. If we had a naturally occurring protein and just randomly made mutations, then this protein would become better and better at its job. That's just extremely unlikely that will be a real sickness. So something was really fundamentally wrong here. And if we can even show that if you just represented a protein just by one feature, which was the number of mutations away from the wild type, then you also got a really good fit. So something was definitely wrong. And the problem is just exactly what I mentioned to you before, that the way that this data was acquired was in iterations, where in each iteration you choose the best candidates, and then from those best candidates, you then make a new round of mutations. So you get further and further away, but in each iteration, you've chosen your candidates to be the best among the previous candidates. So for that reason, we see that as more and more mutations are made, the fitness just goes up, but that's because there's a sort of an extreme distribution shift going on through the selection process over iterations. So we're really not modeling anything relevant here. So once we got the metadata for this, where we had times of when these experiments were conducted, we could do a better split and say, okay, we're going to train up to some time point, and then we're going to test on the next iteration. And if we do that, we get a pretty depressing result, which is that we get Spearman correlations of 0.2, but not only that, we learn nothing over the course of this time. So these experiments, they went on for more than a year, where they're constantly doing experiments, and according to our machine learning model, we're not any better at making predictions at the end of that 1.5 year run than we were at the beginning. So it's very depressing. If we look at the uncertainties, which was the other thing I needed for my surrogate functions to have good uncertainties. If we look at that, that's also pretty depressing, because you would expect that as you go further away from your natural variant, that you would be more and more uncertain about how well this mutant was doing. But our regression methods basically all cannot see the difference between mutations that are close by and mutations that are far away. The only method that could see a little bit of a tendency towards increasing uncertainty was the linear kernel GP. All right, so from that fairly depressing point of departure, what we decided to do was to write a new kernel. So you'll hear about kernels on Friday when we talk about GPs, I'm sure. So we decided to make a very sort of tailored kernel for exactly this problem of regressing on protein variants. And I won't go into the detail because I don't have enough time, but what we did was that we looked carefully. We had both a protein structure and a protein sequence component, and we looked very carefully about if you make a particular experiment on a protein, does that give you any information about other variants that we could have made, maybe at the same position or at close by positions? So we tried to sort of write up what we expected, where we expected sort of information transfer to be possible, and then we made this kernel. And currently this is actually sort of the best prediction for variant effects that's out there. And this is just to make the point that a lot of people will say that GPs are outdated and they're not really very useful anymore. You should just do deep learning. But there are particular settings, especially in the low data regime, where they're still sort of the method of choice, especially if you also want uncertainty quantification. And uncertainty-wise, with this kernel, we also get much nicer properties. And we see now that as we go further and further away, our uncertainty sort of increases, and sort of it has the intuitive properties we would expect from interpolation versus extrapolation and things like that. All right. So that was all I wanted to say. I just want to end with a few takeaways. So the topics that you'll be hearing about this course this week are relevant, not only sort of because you like math and you like probabilities, but they actually have real-world impact. I hope that I illustrated that with a few examples. The first part, I intended to illustrate that modeling choices are really quite important. So how you end up sort of factorizing your probability distributions the choices have impact, and we saw that, for instance, the ability to calculate likelihoods depended critically on how we chose to model the problem. And these modeling things have been going on for decades. Amino acid, there are papers from the 90s about people modeling amino acid sequences, and we're still looking at it today, and we're still making improvements on in this field. And part two, as I said, Gaussian processes are useful, and they're not only academically interesting. And then I never really returned to this Bayesian optimization, which I think still is a super compelling idea, but it really requires high-quality surrogates to work well. And that can be really difficult in high dimensions. And I think we're sort of working hard to try and figure out whether we can get that to work in ways that are systematically better than just doing random sampling. But I think the jury is still a little bit out on whether ultimately that will have a big impact or not. Are there any questions? There was a question. Yes?
Do we have any success stories on protein engineering yet?
The question was whether there are any sort of positive stories on protein engineering. Yeah, so one of the big problems in protein engineering is that typically the labs that develop methods are not the same labs that have access to experimental validation protocols. And this means we very often end up optimizing against sort of computational surrogate, sort of replacements, oracles, instead of actual experimental data. And I think even in that area, we need better sort of benchmarks to really establish whether what works and what doesn't.
The question is, do we need to depend on the table? You know, the protein table.
Yeah, the alignment? Yeah. Yes? Yes?
Do we depend on that to model, or we depend on the data bank? You know,
Yes. Yes. Yes.
and depend on getting our data sets.
Yes.
Modeling. So which of them do you suggest we?
Yeah, so there are, so when I say an alignment, the way it works is that you have a sequence that you're interested in, then you ask the databases for all the sequences that are similar to it. So then you get thousands of sequences or 100 sequences. Then you run an alignment algorithm to get you sort of this nice table. So you use the databases to get this alignment. So that's, and that's if you want to model a single sort of protein family of things that are similar to one protein. If you want to model all proteins in the entire world, then you have to sort of go beyond alignment. So you need to find some other way. And there, these attention-based mechanisms, sort of language modeling type styles, are successful there. But yes, so, and you can also have combinations between the two.
Have you seen large language models being applied to this as well? Because you can see them as the sequences text as well, and then
Yeah, so the question was whether we've seen large language models applied to this, and definitely over the last years, everything is large language modeling in this area in terms of so you have these for generating embeddings. It's now completely standard to use language models for that. The probabilistic interpretation of large language models is a little bit more tricky, so that's why I didn't talk about it today. But it is it's definitely highly used. For instance, for this kernel that I just presented here, the components that go into this are actually embeddings from large language models to get a good sort of idea of where we can transfer between. That's based on considerations from embeddings. So yeah.
the new kernel that you defined,
Yes.
that also provide insight into the underlying biology, so can you use it beyond making this type of optimization?
That's a good question. I think it's primarily a way for us to take pre-existing knowledge and use it sort of actively in a kernel rather than the opposite way around to learn about biology. We sort of it's a way to get prior information in a constructive way. Because the problem is that there's just very little to learn when you only have like hundreds of observations. There's a limit to how much you can actually learn from that, other than sort of this shape of the surrogate function that you're trying to model. So I don't think so, but yes, that's a good question. I'll think some more about it.